Hello everyone. My name's Ying. So I'm the product manager from CUSABIO. For today's webinar, I will show the CAR T-cell therapy with you and how it advanced the personalized cancer treatment. So for today's topic, we will talk about three different parts. The first part will be the advanced research in the current CAR T-cell therapy. And then the second part, we will talk about the key parameters that researchers may need to pay attention to in the CAR construct design, and also talk about the challenge and the limitations that during the research. Then for the last part, we will talk about the emerging landscape and then some future strategies that the researchers may need to be careful in balancing the toxicity and the efficacy. For today's story, I want to start the incredible story of Emily, and also the relationship between Emily and CAR T. So it's a pretty incredible story, and then I think right now is a happy ending. So Emily, she was diagnosed with acute lymphoblastic leukemia in 2010, and at that time she was only 5 years old. So she got several treatments, but then, the leukemia become resistant to the treatment. So there's no choice left for her to last her life. So, and then in 2012, there's a CAR T-cell therapy in the clinical phase 1. And then so she got the treatment, she received the CAR T-cell therapy, and the story right now is a happy ending. So incredibly, the T-cell therapy worked. And then after 10 years, so I think this image comes from the 2022. So at that time, Emily has been like a 10 years cancer-free from the leukemia. So it's pretty incredible story of Emily, but it's also an incredible story of CAR T-cell therapy. So you may wondering what is the full name for the CAR T? So CAR represents the chimeric antigen receptor. So the basic concept behind this CAR T therapy is that at the very beginning is a collection of the blood from the patient. So inside the blood samples, the T cells are separated and isolated. Then this population, they perform gene editing and then transduce with the operate gene to create the CAR T cells. So this population, this engineered cells, they will propagate in the lab, especially the cell expansion. And after cell expansion, the population will reach a titer that can be effective, and then infuse back into the same individual that the T cells come from. And inside the patient, the CAR T cells will take the action and then attack the cancer cells to cure the patient. So, the CAR T cell therapy just represented cell therapy. Actually, the landscape of the cancer therapies are pretty huge. Here is a comparison of different pipelines across the various cell therapies that from 2020 to 2022. I think the overall number here is 2,756 active cell therapy at 2022. And the point that I want here is to show that the change in the cell therapy pipeline from 2020 and then to 2022, the increase is pretty sharp. All in all, I think the number is more than 50%. So you can see that because this big number, so I will say that the overall trend for the cell therapies keeps increasing, and I think it will become more and more. If we take a look at the history of the CAR T cell therapy, you can see that there are some key milestones in the development of the cell therapy. The concept can date back to 1987. So at that time, it was first reported by a Japanese immunologist, Dr. Ishikazu, and his team. And then at 2006, the first generation of CAR constructs comes up. And at the 2011, there's a second generation CAR. And then 2012 is the story from the Emily. And then luckily, 2017, the first CAR T product was approved by the FDA for leukemia. So currently, there are like a total 7 different CAR T products in the market. As we see here, they were approved at different years from 2017 to this year. I think the most recent one was approved last month. If you take a look at the target that for this CAR T product, most of them, they focus on the CD19 and then BCMA. I think the main reason is because these antigens, they are major antigens. And also, if we take a look at the disease, they approved for all of them, they belong to the blood cancers. So, which means that for the solid tumors, the CAR T-cell therapy has a long way to go. So we already see a big progress in the CAR T-cell therapy research. Then the second part, we will check the key parameters that in the CAR construct design and the limitations of the CAR T-cell therapy. The antigen specificity of a CAR is very important. And here I want to bring a concept to you before I talk about the tumor antigen recognition. So first, this is the scFv domain. Is the smallest function domain of the monoclonal antibody. The scFv is the single chain variable fragment. So you see this is the monoclonal antibody. And then this is the smallest function domain of this monoclonal antibody. It contains the variable chain and the light chain. Between the light chain and the heavy chain, there's a fast flexible linker linked these two parts. And the difference between the conventional TCR and the synthetic TCR in the CAR T is that this is the T cells. It recognizes the peptide major histocompatible complex, which we usually call the PMHC. But in the synthetic CAR, the CAR T cells recognize the tumor-associated antigens that on the surface of the cells. So with the scFv module that we mentioned. So scFv module gives the specific recognition between the tumor antigens and the CAR T cells. After the CAR T cells recognize the target antigen in all the cancer cells, simultaneously the interaction will activate the primary domain inside the T cells and also the secondary co-stimulatory signal in the T cells. Then after that, the cytokine will be released and also will proliferate to perform the cytolytic activities to kill the cancer cells. So this is the whole progress from the recognition to the killings. I mentioned that I will talk about the key design parameters, and here are the summary of the 4 different parameters that researchers care about during the CAR construct design. So they are the single chain variable fragment, the scFv module, and the spacer domain, transmembrane domain, and the cytoplasmic domain. For the single chain variable fragment, its function is in the antigen binding activities. And for the spacer domain, is connecting the scFv module to the transmembrane domain. The third transmembrane domain is aiming to transmit the ligand recognition signals to the cytoplasmic domains within the cell. And then after the signal was transmitted to the cytoplasmic domain, this domain will kickstart the signal cascade and then to release the cytokine and then to start the later response. Here, let's take a closer look at the CAR construct. First, the single chain variable fragment module. For this part, I think most of the researchers, they care about the affinity and avidity of the ligand binding domain because the affinity is pretty important, and it's like increase the affinity is to improve the specificity of the CARs. So after you increase the specificity, you can reduce the on-target of tumor side effects. And this is really important if your target antigen is ubiquitously expressed on the healthy tissues. And I think it's pretty common to see that in the solid tumors, the antigen is widespread, expressed on the healthy tissues. So antigen selection and the affinity is very important to reduce the toxicity. And also another thing is about the mispairing or aggregation of the single chain variable fragment. For example, if the scFv module, they aggregate together, then it may cause a pretty high expression level on the surface. Then it can cause the excess tonic signaling, and this excess signaling will cause the early exhaustion of T cells. Then it will reduce the efficacy. And the last point about the scFv module design here is the flexibility. So you see here, we always want to target the epitopes on the surface of the cancer cells. But you can imagine some of the epitopes, they are pretty difficult to target. So we want to have a better recognition. And one example about the difficult epitope is antigen called the MUC1. So MUC1 is heterogeneously glycosylated molecules. So it's kind of like in the design, the scFv module design need to be flexible, especially for this one. The second parameter is the spacer domain. The main design related to this parameter is to lend the flexibility to the scFv and then to help the improve efficacy. In a simple word, is that we want to have a spacer domain to enable the recognition of the target epitopes and then to have a better recognition. And this better recognition can give a profound effect on the activity and the signaling of the CAR T cells. Then the third one, I think, is also can relate it to the better recognition, because if we think about in the real world, in our physiological T cells, the transmembrane domain of the TCR-CD3 complex, they are pretty difficult. And actually, the signaling mechanism of this complex has not been fully elucidated. So we are kind of like unknown to what is the real situation there. So we want to have a different CAR designs to mimic the real world, because usually in the CAR design, the structure is pretty linear, so it's not that 3D structure. So with this novel CAR designs, we are kind of like potentially to build a more effective physiological signaling profile and then try to mimic the real world. So I think people are trying to like different CAR designs to try to get the things work out. Then the last one, I think, is also very important because the cytoplasmic domains, they are highly related with the signaling. So as you see here, in the first generation CAR, here we only have a functional cytoplasmic domain, and then it can provide an activating signal after the ligand engagement. But as you see, after, for the second generation and the third generation, you see the number of the cytoplasmic domain increased. Except the primary signaling, we also see the co-stimulatory signal. Let's call it signal 2. So the signal 2, the function is to magnify the signal. And then luckily, the second generation and the third generation, the results are good. They have been tested against different targets, such as the CD19, the PSMA, and the GD2, and they worked. So it seems like the co-stimulatory domain is pretty important. And in summary, in the current clinical trials, I saw that the designs, they always combine the signal 1 with the signal 2. So signal 1 is the initial primary signal, and then signal 2 is the magnifying signal. So they are pretty common in the next 2 generations, and then you will see them in 1 minute. So overall, for the key designs, the concept here, I will say each component, they contribute towards the proper activation, functionality, and the persistence of CAR T cells. And we talk about the 3 generations, right? And if you see the 4th generation, there's a novel design here. So here they put inducible express the construct here. With this inducible express the construct, the CAR T can express the cytokines. So it's kind of like inducible to limit the toxicity. And also for the 5th generation, you see the cytoplasmic domain, they have an additional motif to provide the antigen-dependent cytokine signaling. So overall, these 2 designs, they are trying to increase the efficacy and then try to limit the toxicity. But let's take a look at all the 5 generations. You can see that the SCFA is well conserved, and then the cytoplasmic domain is pretty conserved. So these 2 parts are important, and then they are usually well preserved. After I talk about the design parameters, here I want to give you a summary of the killing mechanisms after the CAR T cells recognize the tumor antigens. So basically, they can be classified into the CAR-dependent and the CAR-independent cytotoxicity. So for example, the first one, for the antigen-positive tumor cells, inside the native T cells, the main killing process is that they enter and release the cytotoxic effector proteins. So like the peripherin and the grain enzyme. So these 2 proteins, they will perform the granulars inside the cells. And the performing, it will introduce the holes inside the cell membranes. Then with the holes, the grain enzyme can enter into the target cells and then induce the apoptosis. For the other one is the death receptor pathway. This one requires the membrane-bound Fas ligand. So this ligand expressed by the T cells, right? And it will bind to the Fas on the target cells and also induce the apoptosis. So these 2, the peripherin grain enzyme pathway and the death receptor pathway, these 2 are major killing pathway. And then to a lesser degree, there's another pathway called the cytokine pathway. This cytokine pathway is rely on the cytotoxic T cells. This kind of T cells, they will release the different cytokines. For example, the TNF alpha and INF gamma. These 2 cytokines will lead to the increased target cell recognition and the killing. So overall, you see that these 3 different killing mechanisms contribute to the apoptosis of cancer cells. Then after the killing mechanism, here I want to give you a summary of the potential hot targets, not only in the approved CAR T, but also in the clinical trials of our CAR T product. And you can see a very clear difference between the liquid tumor and the solid tumor. For the liquid tumor, we talk about the CD19 and the BCMA because they are in the approval CAR T product, but some other like the CD22 and the CD20, they are very often found in the clinical trial. And for the solid tumors, there are also some mature that you can find. They are highly correlated to the breast cancer, lung cancer, and then pancreatic cancer and ovarian cancers. So for example, like the MUC1, HER2, GPC3, and EGFR. So this kind of like a target, they are all maturation antigens that abundantly expressed on the cell surface comparing to the others. Similar to other therapies, I will say that researchers, when they design the CAR T construct, they are always pursuing a balance between the efficacy and the toxicity, because we always need to consider that in the clinical trial, how the patients suffer from. We always want to increase the efficacy, but always want to decrease the suffering that the patients, there. So here in this part, I will spend some time to talk about limitations and then challenges in the clinical trial and how the researchers trying to avoid the toxicity, how they can increase the efficacy. The summary here includes the antigen escape on target of tumor, CAR T-cell trafficking and the infiltration, immunosuppressive microenvironment, and the last is the associated toxicity with the CAR T-cells. So I kind of like group them together. For the first aspect, I think the limitation, they are highly correlated with the antigens. So for example, current clinical trial, I think majority of them, they only have the single antigen they belong to the single antigen targeting CAR construct. But for this kind of like a CAR, it's so easy to develop the tumor resistant. And actually a large portion of the patients, after they receive the treatment, they display either partial or complete loss of the target antigen expression. This may happen because the antigen mutated or some alternative splicing of the antigen. And then this will result like the efficacy decreased and also the relapse of the disease. And another is the on target of tumor. This one is highly related to the solid tumors because for ideally antigens, we want the antigen extracellular expressed on the surface, and then they are easy to be accessed. And also another one is that we want the antigen to be uniform or consistent expressed on the malignant cells, but not in the healthy cells. But you see, it's an ideal situation. Actually, in the real world, it's not that the case. Usually the antigens that they not only expressed on the malignant cells, but also expressed on the normal tissue cells, sometimes with the varying levels. So overall, the antigen selection is really, really important. And I think this is the first thing that researchers need to consider at the very beginning when they start the design. And the second part, I think the limitation is also close related to the solid tumors. Because with the liquid tumors, I think it's easy for CAR T cells to access, either directly in the blood or via the lymph system. But in the solid tumors, things changed. CAR T cells, they recruited into the solid tumors, but around the tumors, they will encounter many other cells within the microenvironment. And in the microenvironment, there are physical barriers that will block the CAR T cells to enter into the tumor cells. Thinking about your tumor cells, at that site, there's always very dense extracellular matrix there. And this extracellular matrix will trap the T cells so they cannot get in. And also around the environment, there was some checkpoint inhibitor there, like from the PD-1 or the CTLA-4. And also there were some other soluble inhibitors in the form of the suppressive cytokines, for example, the IL-10 and IL-6. So these two, they will also disrupt the T-cell effector function. Even the metabolic environment around the tumor, they will disrupt the effector T-cells function, like because there's a lack of the nutrients there and the condition is pretty acid, and then also there was some toxic byproduct. All this, they will disrupt the function of the CAR T cells, then decrease the efficacy. The last is the limitations that from the associated toxicity of the CAR T. After infusion of the CAR T cell product, I think like 80% of the patients, they are suffer from the CRS and the ICANS. So usually the toxicity from the cytokines, including the IL-1, IL-6, IFN gamma, and the TNF alpha. Because the CAR T cells is activated, right, within the tumors, then they will induce some cell activation. the interaction between the CAR T cell and the myeloid cells, they will create some excess circulating cytokines. And these excess cytokines, they will contribute to some activation, induce the blood-brain barrier dysfunction. This blood-brain barrier dysfunction will keeps enter into the central nervous system. So after the CRS, it's always very easy to see that the patients all have some neurotoxicity associated toxicity. So, it's pretty symptom that observed in the clinic. So I think these two types of toxicity is really need to be considered like at the very beginning when you design the CAR T. We talk a lot about the challenge and then the limitations that in the design, but additional consideration should be also focused on the CAR T cell manufacturing. Generally, the CAR T cell manufacturing, it involves the cell isolation, T cell activation, and the transduction, and the expansion, and then finally the formulation. In certain cases, especially the product failed, CAR expression can be the cause. When I say the CAR expression, I mean that after the viral transduction, the positive rate of the CAR. So it's really important to test the positive rate after the viral transduction. Relevant regulations, they already set a very clear requirement for this kind of testing. So it's really crucial to develop accurate, simple, and universal CAR reagent to perform this kind of assay to meet the requirement from the regulations. So regarding this, CUSABIO has already developed the efficient universal CAR detection reagent, which we call the anti-CAR linker monoclonal antibodies. So this kind of antibodies, they are designed to perform this kind of positive test. So they are not restricted by the antigen specificity, but they have a specific binding to the linkers. So if you remember, at the beginning, I introduced the single chain variable fragment. In this module, the flexible linker linked the light chain with the heavy chain. And our monoclonal antibody recognized the linker site here. Commonly not only in the approval CAR T product, but in most clinical trial product, G4S linker and Whitlow linker. These two type of linkers, they are widely used. So our product are corresponding to recognize the G4S linker and also to recognize the Whitlow linker. So as long as the designed CAR constructor have the linkers that I mentioned here, our anti-CAR linker monoclonal antibodies can be used to perform the positive test. And then for this kind of reagent, unlike the others, they can avoid the false positive result. For the CAR linker antibodies, they have a very broad applications. They are pretty suitable for most of the CARs in the market. And also, in addition to the CAR detections, they can also be used for the CAR cell preparation, like the activation and the CAR enrichment. And also, they can be used as reagent to perform the PK analysis and some other following functional tests. So, there's a wide application for this kind of antibodies. So our CUSABIO monoclonal antibodies, they have been verified with the binding activity. For both of the type of the product, we perform the ELISA experiment to test the binding activity, but they can also be directly used in the flow cytometry. So here's the flow cytometry data that we test and validated. The green is the untransfected, the 293F cells, and the red is the transfected cells that containing the anti-CD19 CAR construct. In that construct, we have the linker there. So as you see, the red line, they shift right after staining with our monoclonal antibodies. So So you can always trust us the activity. We verified the binding activity not only with the ELISA, but also with the flow cytometry. So I already talked a lot about the CAR T. However, the CAR T, they must remain within the therapeutic window. I mean, the balance between the efficacy and the toxicity. So some other types of the cell therapies, they emerged and as alternatives or some additional options for the patients to choose. And hopefully with some less alternatives, the patients can get better treatment. Some of the therapies that I want to highlight, one is the CAR Treg, and another one is the CAR NK cells. I will talk them in a little bit details. So the first is the CAR Treg. Treg is the regulatory T cells. These cells, they are subpopulation of the T cells. They can suppress the function of conventional T cells and some other immune cells. So in the past, they already have some therapies that with the Tregs being carried out in the transplantation and autoimmune disease, not only in the animal models, but also in the clinical trials. But at that time, the use of the Treg, is a large number of the polyclonal Treg. So with the polyclonal Tregs, the antigen specificity are unknown. So because this unknown antigen specificity, at that time, they lead to some unwanted effects. But right now with the CAR, the strategy is to engineering the Tregs with the CAR. So instead of using the polyclonal Tregs, they are antigen specific Tregs now. So this type of the therapy is pretty promising in the cancer research. And another thing I want to highlight is that this kind of the cell therapy can become a universal CAR Treg therapy. And the researchers have recently tried to develop this kind of like a universal therapy instead of the personalized therapy. Another therapy I want to talk is the CAR NK. NK represent nature killer cells. Compared with the CAR T therapy, this one has a unique advantage and some characteristics. First one is that the cell source. Because the NK cells, they have a low risk of the rejections. So the cell source can not only just be T cells, they can originate from different cell sources, like the spleen, the liver, the secondary lymphoid organs. So you can think that if at the very beginning, if you start with a large number of the functional NK cells, the success rate can also be increased. And for NK cells themselves, they have a rapid and a potent cytotoxicity. So the CAR therapy comparing to the CAR T will also be pretty strong. Last part, there are two future strategies provided to improve the efficacy and then to decrease the toxicity. For the efficacy, one thing I want to highlight here is the T cell exhaustion. It's pretty common in the CAR T cell. In the T cell exhaustion, the T cells gradually lose the function after the persistent tumor stimulation. So, persistent tumor stimulation, at that time, at this case, the inhibitory receptors, it increased, and then they go back to suppress the T cells function, then the efficacy decreased. Right now, researchers have been identified different mechanisms of the CAR T cell exhaustion happens. So hopefully with this identified mechanisms, they can develop some new strategies to solve this problem. And the last thing about the toxicity is the CRS toxicity that we mentioned. Most of the toxicity because they come from the cytokine. So I think the one strategy is to construct an inducible system to kind of like inducible anti the IL-6 or the IL-1 beta activation. So hopefully by block the activation, it can be helpful to reduce the CRS toxicity. So you see, you see, like not only for the CAR T, I think for most of the treatment that in the world, we care about the toxicity and efficacy. So the balance is obviously important. At last, please give me some minute to introduce our company. We are CUSABIO. It's kind of like a customer-satisfied biological reagent. And then we are specialized in the transmembrane protein expression and antigen selection. So CUSABIO will always be your good partner in biology. For our product, we have 5 different lines, and they contain the ELISA kit, proteins, antibodies, molecular biology product, and the small molecular antigens. Our product has been exported to more than 80 countries, including the US, Canada, Europe, Russia, Japan, and Germany. And we have a very good relationship and cooperation with some big companies like the Pfizer, Sanofi, Bayer, Zinker, Johnson & Johnson. So our product has been widely spread. And lastly, thank you so much. But here I want to remind you that if you are interested with our anti-CAR linker antibodies, please scan this barcode and then apply for a free trial. And then we are happy to help with your research. Thank you so much.